Hi everyone, this is Miss G. These are your class notes on experimental design. This is just a picture that gives you the general flow for how we conduct research in science. We first ask a question, we do some background research on that question, construct a hypothesis from that background research. We're going to test with an experiment, collect data, analyze the results, draw a conclusion. If our hypothesis is found to be supported, then we report the results. If it's not, then we go back and reconstruct a new hypothesis. So our first major step is to come up with a question about a particular topic, and it's got to be a question that's actually testable. So for example, a testable question might be, what is the effect of sugar on the heart rate of mice? A question that would not be testable would be something like, why do mice like sugar? Once we have the question established, we want to do some background research. First of all, we might want to know if the problem's actually been studied before, and if we can get enough information to come up with an informed hypothesis. We don't want to just make a random guess about what's going to happen. Then we'll establish an independent variable. This is sometimes called the manipulated variable. For our experiments, there's only going to be one independent variable. It's got to be very clearly defined, and there have to be different levels of this variable. Notice how the word independent begins with the letter I. So I remember the independent variable is the one that I control. So in my mouse example, our independent variable would have been the sugar. What is the effect of sugar on the heartbeat of mice? I am controlling the amount of sugar. The different levels would be different amounts of sugar. Maybe I'm giving them half a gram, 1 gram, 2 grams, etc. That would be different levels. The dependent variable is going to be called the responsive variable. This is the variable that we're going to observe and measure to see if it in fact changes in response to us deliberately changing the independent variable. So in my mouse example, the responsive variable would be the heart rate of the mouse. When we design experiments, we're going to want to give them very specific titles that actually identify both the independent and the dependent variable. This is what our titles are going to look like. The effect of the independent variable on the dependent variable. So for my mouse example, the title of my experiment would be the effect of sugar on the heart rate of mice. My hypothesis is going to be a possible explanation of the experimental problem, and I only develop a hypothesis after I collect some background research. A hypothesis is not a guess. It's a explanation that's based on some information. The hypothesis also has a format. It's going to be written as an if, then, because statement. So I should see those 3 words in the hypotheses that you write. Again, it's based on research. So in my mouse example, I might say something like this. If increasing levels of sugar are given to mice, then the heart rate of mice will increase because it's been found that sugar increases blood pressure in mice. Constants are factors in your experiment that don't change, so they're kept the same in every single trial. Ideally, the only variables that should change are the independent and dependent variables. When you design an experiment, you should define and identify your constants. So in my mouse example, some constants might be the type of mice I used, the size of the mice, the type of sugar used, etc. The control is a group or subject that serves as a standard for comparison. Not all experiments are going to have a control, but when possible, we'll try to have one. So in my mouse example, the control might be looking at mice who haven't eaten any extra sugar. Here's just a little comic for you. Okay, then we're going to start to design our procedure. The procedure should list any materials that we need. It's got to be descriptive enough so that anybody else in the room could repeat your experiment exactly. Ultimately, when we come up with our final reports, it's going to be written in third person past tense. We're going to involve multiple trials, ideally 5 or more, a minimum of 3. And it should include some kind of label diagram if the setup is complex at all. There are 2 types of data you're going to collect. There's qualitative data and quantitative data. Qualitative data is just descriptive. What do you see? What do you smell? What do you hear? The quantitative data is going to be measurable, so it's going to involve some type of a number. So in my mouse example, the measurable data would be the actual heart rate, whereas the descriptive data would be maybe what the mice are doing. The conclusions that you write are always going to address your original questions, so I would suggest that before you even write your conclusion, go back and look at the question that you wrote down. The conclusion is going to use the results of the experiment to determine if your hypothesis is supported or not. Just remember that hypotheses are never wrong, really. They're just supported or not supported. It's really important that you use evidence in order to determine if your hypothesis is supported or not. So use your data and talk about that when you write a conclusion. I'll talk much more about this later, but we will be writing error analyses as part of your lab reports. And the error analysis will really address factors in the materials or the procedure that might have had an impact. And we're also going to look at how we could improve our experiments. The error analysis is also, if possible, going to compare known values with the values that we received for an experiment. And we'll calculate a % error if possible. We're going to discuss 3 types of errors in our error analysis: instrumental errors, method errors, and human errors. Again, I'll talk about those later. What we want to avoid is writing about personal mistakes. And that's it. So hopefully you wrote all those notes down. Write down any questions you have, and I'll address them in class when I see you next.
Made with Doodlecast Pro.